Next speaker is Dhani from University of Adelaide, all the way from Australia. And she will talk about understanding emergent dynamics in AFib using cardiac models, AI, and EQ.
Good afternoon, everyone, and thank you to the event organisers for the privilege of being able to present this work today. So just to preface, this is probably going to be a little bit less technical compared to some of the presentations on similar methodology presented today, but I hope that it can provide sort of an overview of a potential application for some of these technical approaches. And specifically, that's to understand some emergent dynamics in atrial fibrillation. So to preface, and some of this might be very trivial to many of you, the concept of emergence really relates to complex patterns or behaviours that arise due to the interactions of simpler elements within that system. So this can be used to conceptualise things like the way cells coordinate and specialise to form tissues and organs in biology, the interactions between animals to form ecosystems, and interactions similarly between people to form social structures. And oftentimes these emergent patterns, behaviours, or structures are not immediately predictable and understandable just from the individual elements in that system, but have to be understood through those complex interactions. And so in a similar way, in AF, emergence can be seen in how cardiac cells in the heart might interact complexly to produce electrical excitation waves and turbulent electrical activity like you would see during an arrhythmia like atrial fibrillation. And this might not, again, be necessarily predictable from the individual behaviours of those cardiac cells. So if we take, for example, a singular cardiac myocyte, we might be able to look at the action potential dynamics of that singular cell, but then when they aggregate and kind of form together this tissue, then they have much more complex interactions, which lead to these more complex excitation waves and turbulent wave dynamics during arrhythmias. And although this kind of covers the concept of cardiac electrophysiology and things like the formation of reentrant circuits and spiral waves in AF, this can also be used to conceptualize electrical and structural remodeling and atrial stretch and enlargement. However, we were kind of most interested in this emergent behaviour in cardiac electrophysiology. So as part of a multi-centre clinical study currently being run across 4 Australian hospitals, we have been investigating some of these emergent properties that we observe in clinical episodes of atrial fibrillation, and more specifically from routine clinical EP data that is taken for patients clinically indicated for AF ablation. And so we were most interested in looking at things like how to characterize this turbulent wave dynamics and also electrical organization in the atria as well, which in some instances can evolve and show some differences, especially in cases where, for example, atrial fibrillation might self-organize and terminate. And so just very briefly touching on some of the previous work, we looked at some of these kind of bespoke metrics that we tried to develop for capturing some of this emergent behaviour. And the first of which that we investigated was this measure of correlation length, which is used in sort of other disciplines like physics, where we take a look at the distance over which activity essentially remains correlated or similar. And so in this context, we look at the correlation of electrical activity, which was estimated here by the relationship between the pairwise electrode combinations that we get, and calculating the two-point cross-correlation coefficients from that, and then plotting that against the relative electrode distance, which gives us this kind of characteristic exponential curve in which we can then approximate a correlation length. And so you can imagine if we have sort of more organized episodes of fibrillation where there's more organized electrical activity, we might get more higher correlation lengths, and conversely, if we have more disorganized episodes, we might have a lower correlation length. And then additionally, we also had looked at trying to establish these renewal rate metrics, which tries to capture some of the properties to do with turbulent wave dynamics, but namely associated with the formation and destruction of spiral waves and wavelets. And so an analogy for this was it's rooted in probability theory and statistics, where, for example, in thermodynamics, you might have temperature of a gas, which is related to the average kinetic and energy or speed of molecules in that gas. However, the molecules don't always have the same speed, even if all the gas is all at a single temperature. And so what is typically done is you take the distribution of the molecule speeds at certain temperatures, and that's a way of connecting the individual molecules to the emergent properties of that system, which in this case is temperature. And so borrowing this statistical framework, we essentially applied this to try to study the formation and destruction of spiral waves and wavelets, which were defined as by the phase singularities, which occur at the pivoting regions of these spiral waves and at the free ends of wavelets. And we essentially looked at the times between new consecutive phase singularity formations and constructed a distribution of those inter-event times, and then similarly looked at the lifetimes of phase singularities and then constructed a distribution of those. And so what that allowed us to do was characterize the average rate of formation, these phase singularities denoted by lambda f, and destruction denoted by lambda d. And so why are we interested in these potentially kooky metrics? One, not only are they potentially useful to try to capture some of these emergent properties, but also we've been investigating through our clinical study the association of these metrics to clinical outcomes and endpoints. So some work being done by my clinical colleagues have shown some association to AF termination and clinical AF phenotype, as well as some response to antiarrhythmic medication. And more recently, the association to time to AF recurrence following AF ablation, or in other words, a time or the duration of sinus rhythm following AF ablation. And so we have these metrics, but for them to have any real clinical utility, there still stands this underlying question, and that is, what are the determinants that govern and influence these metrics? So for them to be able to be used as targets in an intervention, it would be really useful for us to understand what kind of drive these metrics, because then we would be able to understand, for example, if a particular type of antiarrhythmic therapy might be useful to guide some intervention strategy based on these metrics. So this kind of segues into this more recent body of work, which is fairly preliminary, so I apologize, but it takes a lot of inspiration from work being done by others in this space, and we've heard a little bit about Gaussian Process Emulators, or GPEs, today, from people like Steve's group and Richard's group as well, and essentially the idea was to try to use GPEs to map the relationship between various physiologic and cellular parameters in cardiac models to these clinical metrics. And as this initial pilot study, we really just looked at a reduced parameter set, which is just the ion channel, a few ion channel conductances and tissue conductivities. And so to simplify this problem, just as an initial proof of concept study, we looked at just some 2D square grid simulations, which represented a 4 centimeter by 4 centimeter tissue slab. And this was just trying to see if this had any application for this type of problem, and whether we could use this framework to understand the relationship between the different model parameters and our metrics. And we looked at this in 2 different cardiac models, the Courtemance model and the atrial adaptation of the Luo-Rudy model, with the idea that these kind of give 2 different flavors of electrical kind of patterns or behaviors, with the Courtemance kind of depicting this more stable spiral phenomena, and the Luo-Rudy having more of this multiple wave-like behavior. And so we focused on these initial ionic and tissue conductivity parameters as shown here, again just as an initial proof of concept validation. And then to explore this, we generated 200 simulations using Latin hypercube sampling to select uniformly random combinations of these model parameters. And the methodological overview was essentially as follows. So we have these 2D simulations, and then to have them reflect the clinical recordings more closely, we then looked at the electrograms and constructed a virtual catheter that would be representative of the clinical catheter that we map with, and then calculate the AF metrics on these virtual catheter measurements, and then perform a global sensitivity analysis using the GPE. So to construct the virtual catheter, we tried to essentially mimic the HD grid catheter, which is the one that's used for our particular clinical study. And so the way that we tried to construct this was as follows. So given that the HD grid is this kind of 4x4 grid-shaped catheter with a 3mm inter-electrode spacing, we were able to calculate the node spacing and the electrode spacing that would replicate the spatial resolution of the catheter, and then calculate the number of unique catheter positions that we would have for the virtual catheters in our 2D grids. However, we have about 16 different virtual catheter measurements for each simulation, so that presents a potential problem, and that is for an already potentially high dimensional parameter space, that adds even more complexity. And that ends up being not dissimilar to what we have for our clinical recordings, because we essentially have 16 different HD grid measurements for each individual patient because of the way that the recordings are taken across 16 different anatomical locations. And so more work needs to be done here, but just as a very preliminary simplified approach, just to check whether this GPE framework was viable here, we just implemented a very simple subsampling approach by taking every kind of third catheter measurement, and this was facilitated by the fact that there's no temporal variation in when the virtual catheter recordings can be taken here in the simulations, which helps simplify things. But currently we are looking at more sophisticated approaches for potentially reducing dimensionality and also better subsampling virtual catheter measurements. So again, this is very preliminary work, but in terms of just initial findings, so we evaluated both the Courtemance and Luo-Rudy models over 100 epochs, which was what we found to be the sweet spot to ensure sufficient training without starting to overfit the model. And we essentially used the GPE to evaluate the model, each of the metrics separately, so we could get an idea of how each of how the model parameters are affecting each of these metrics individually. And so we can see that there is still room to go. There's only modest performance with the GPE. So with the Courtemance, you can see that there was an R squared of about 0.62 for the correlation length, and then respectively 0.66 and 64 for lambda F and lambda D. And then for Luo-Rudy, it's slightly better with a 0.78 for the correlation length and 0.69 and 0.67 for lambda F and lambda D respectively. So it might suggest that the GPE framework as it currently stands is still not able to fully elucidate the complex relationships between the model parameters and these metrics, but we still have some optimization to go. There is also some suggestion that the Luo-Rudy model might have some better prediction accuracy and generalization, but again, this can be improved. And then when we looked at the sensitivity analysis, we saw some trends. So in relation to the correlation length given by psi, the inward rectifier potassium conductance and longitudinal tissue conductivity seem to be the most influential to the degree of electrical correlation or organization, at least when measured by the correlation length. And similarly for spiral wave formation or lambda F, there was highest sensitivity to these metrics as well, but also this low inward calcium conductance in the Luo-Rudy model. So that might indicate that these have the most influence on phase singularity formation, which may be perhaps due to controlling excitability and wavefront propagation. And then similarly, we had this same trend, highest sensitivity to these model parameters for the destruction rate. Again, perhaps by way of governing the repolarization and recovery processes. So a few, I guess, caveats to mention here for this initial pilot. Again, there's still scope to further explore this area, but there is this issue of being able to encode that spatial variability. And so that's not only evident in our simulations, but also real clinical data as well. In addition, in the clinical measurements, there is a lot of temporal variation in the catheter recordings because they are taken sequentially rather than simultaneously. So we might need a way to encode some of this temporal information as well. Obviously, performance is modest, and so we need to likely further optimize the hyperparameters in the kernel that's used by the GPE. And then we've only really looked at a few bespoke AF metrics as far, which may not have easy to capture relationships to the model parameters that we investigate. And then finally, simulations that we use might not actually fully capture the heterogeneity and stochasticity and ionic and tissue conductivities that are present in real patients. And so some further directions, we need ways to still determine the most viable sampling or dimension reduction technique, potentially explore other model parameters for a more rigorous sensitivity analysis, which has been done, for example, by others here. Also potentially exploring alternative EP metrics for other emergent properties, which we're actively exploring. Scaling to obviously more sophisticated anatomically accurate digital twins, which have been well established by other groups. And then potentially also exploring other cardiac models as well, in addition to the Courtemance and Luo-Rudy models. Thank you very much.
Time for a couple of quick questions. I guess I can start one. So it's very interesting to see those EP metrics and to see that they are correlated with the clinical outcomes. Can you elaborate a little bit? I was sort of curious about the benefit of introducing those metrics. Like, can I do GPE or sentinel analysis directly from the parameter space to the clinical outcome of interest instead of introducing these metrics in the middle?
Yeah, that's probably an interesting question. So I guess the reason we were motivated to specifically explore those metrics is to kind of tie into our previous work where we established those metrics. So I guess it was a pragmatic extension of previous research work, but also the fact that we can calculate these types of metrics from routine clinical EP data, and so we think that could be potentially advantageous because it's much harder to try to calculate some of these other metrics that oftentimes are used in to validate and calibrate cardiac digital twins and models as well. And so we kind of thought it would be a useful thing to try to, I guess, investigate these EP metrics in that way. And then that opens this, I guess, scope for if other groups develop similar bespoke metrics, perhaps they can utilize a similar framework as well. In terms of, I guess, using the GPE to directly look at relationships to the clinical endpoints rather than introducing the metrics in the middle, that's a really interesting question and a great one, and I hadn't thought of that, but probably useful to think about that as well.
I guess just to follow up on that, so you mentioned that those metrics are relatively easy to obtain in clinical practice. So what are the requirements? Like, what type of data do you need? And in terms of quality of the data do you need in those catheter mapping in order to derive these metrics reliably?
Yeah, so I guess easy is relative somewhat, but I guess they're readily measurable from clinical EP data. So just from taking the electrograms, we can essentially just apply signal processing approaches that are well established in literature to measure these metrics. So one thing that we're actually currently investigating is we've established this real-time software platform which essentially builds upon Abbott's LiveSync system capability so that we can actually measure some of these metrics in real time as well. And so that has been quite useful to get more nuanced insights into how these metrics change as well, particularly in response to antiarrhythmic interventions, because we are seeing some emergent behaviours that are not immediately, I think they weren't immediately very obvious from just looking at the export before.
Yeah, it's pretty cool to be able to see them in real life. Yeah, you were time.
Yeah, the total order indices look really high. So I was just wondering if you had any perspective on the first order indices to try and make some deductions about interactions or anything like that.
Yeah, I haven't included that here, and again, this is very preliminary, so there's a lot more I need to explore, I think, in terms of first order and looking at rather than the total effects, because maybe there's something useful to elucidate there. But I think this approach I've learned a lot today in the use on the use of GPEs, I think, and so I hope to kind of build that into a more rigorous framework for using this approach.
Thank you. I thank our speaker again for